Welcome to Inert Chemistry. Today's video lecture we are discussing the part 2 of biosynthesis of purine nucleotide. In the first part we see that how inosine monophosphate is formed. Inosine monophosphate is a parent nucleotide and all the other nucleotides are formed from inosine monophosphate. Here first we see that how adenosine monophosphate is formed from inosine monophosphate. This is the structure of inosine monophosphate. Here you see this is ribose 5-phosphate and on ribose 5-phosphate this inosine monophosphate is formed. So first of all aspartate and GTP are utilized. They are incorporated into inosine monophosphate and the resulting compound is adenyl succinate. Here you see that this aspartate group is added into the inosine monophosphate. The reaction is catalyzed by enzyme adenyl succinate synthetase. This adenyl succinate synthetase, it is an intermediate, and after that fumarate molecule is released from this by the help of enzyme adenyl succinase, and at the end adenosine monophosphate is formed. This adenosine monophosphate is further converted to adenosine diphosphate and adenosine triphosphate by the help of enzyme adenosine monophosphate kinase and adenosine diphosphate kinase respectively. In these reactions, the phosphate group donor is ATP. Synthesis of GMP means guanosine monophosphate from IMP. Again, this GMP is also formed from inosine monophosphate. NAD plus is used and NAD is converted to NADH and xanthosine monophosphate or XMP is formed. This is an intermediate and later on glutamine is used. Glutamine donates the amino group and converted to glutamate. In this reaction ATP molecule is also utilized and xanthosine monophosphate is converted to guanosine monophosphate by the help of enzyme guanosine monophosphate synthetase. Guanosine monophosphate is further converted to guanosine diphosphate and guanosine triphosphate. Formation of deoxyribonucleotides diphosphates. In the previous slides, we see about the synthesis of ribonucleotides. But here we see the synthesis of deoxyribonucleosides. Deoxyribonucleosides are formed from ribonucleosides. For example, ADP and GDP, they are converted to deoxy ADP and deoxy GDP by the help of enzyme ribonucleotide reductase. This reaction requires reduced thioredoxin that is oxidized during the reaction. To convert the oxidized thioredoxin back to reduced thioredoxin, thioredoxin reductase enzyme is used and NADPH molecule is used. Remember that both these ribonucleotide reductase and thioredoxin reductase complex, it is functional only when cells are actively synthesizing DNA, means in the S phase of cell cycle. Here is the summary of all these three slides. Means here is this is inosine monophosphate. It is converted to adenosine monophosphate, and similarly inosine monophosphate is converted to guanosine monophosphate. This is all about the biosynthesis of purine nucleotides. In the next video, we will see about the regulation.